Hello, my name is Quintero. I am a part of Dr. Lisa Hua's lab here in the biology department of Sonoma State University. I am proud and honored to be a COPLAC presenter, and I am very excited to be presenting my research project on the positioning of homologous chromosome 15 in Bloom syndrome. I wanted to start off with a little bit of background. Here in white is a cell, and within that cell are two red chromosomes, and as a reminder, we have two sets of chromosomes, and the relationship between each chromosome in those two sets is homology. So in black and white, I provided alleles, which are DNA coding regions that are the same genes, but they are not necessarily identical, hence the two different colors between those two chromosomes. And what you can see is that along both of the chromosomes, they will have alleles in the same position, but in different colors, and that shows homology. And this shows that these homologous chromosomes are structurally the same, but not necessarily identical. And I would like to introduce you to meiosis, which is sex cell division, and specifically in prophase 1, there's a hallmark called crossover. And crossover is where the homologous chromosomes will pair right next to one another and go through crossover, which is reciprocal exchange of DNA between the homologous chromosomes, which I showed with the little arrows, where the alleles are swapping between the two chromosome arms. And what this ultimately results in is in 4 genetically diverse daughter cells. And this promotes genetic diversity. In contrast to mitosis, which is non-sex cell division, what you find is the homologous chromosomes are actually spatially separated, therefore they do not go through crossover. And what this ultimately results in is in genetically identical daughter cells. So I'm investigating how chromosome 15 will organize within Bloom syndrome cells. So for a little bit of background, Bloom syndrome is an autosomal recessive disorder characterized by sunlight sensitivity, stunted growth, immunodeficiency, and predisposition to cancer. And at a microscopic level, what they found is there are implications where chromosome 15 does abnormally go through mitotic recombination. However, it is unknown whether chromosome 15 actually does pair. And once again, mitotic recombination is abnormal because in mitosis, as a reminder, the homologous chromosomes should not go through recombination. It should be spatially separated to create genetically identical daughter cells. And so I hypothesize that homologous chromosome 15 within these Bloom syndrome cells will pair. In contrast, I could also see a different finding where homologous chromosome 15 actually is spatially separated, therefore they do not pair. The way in which I would like to test my hypothesis is by first culturing Bloom syndrome fibroblast cells in vitro on these culture plates with slides on them. And once I grow enough cells on these slides and fix them or preserve them, what I would like to do is perform fluorescence in situ hybridization, otherwise known as FISH. And the mechanism of FISH is that we take advantage of the fact that each chromosome is containing sequence-specific regions of DNA that are unique to each chromosome. So we would like to investigate chromosome 15. So what we do is take these probes that are analogous to these sequence-specific regions of DNA on chromosome 15, and we will denature the DNA in these cells, essentially that double helical structure of DNA. We will unwind it and unzip it, so now that the probes could hybridize and bind and have affinity toward that sequence-specific part of chromosome 15. And for the rest of the DNA, we'll apply a DNA counterstain. And the cool thing about this FISH is that we have the probes for chromosome 15 fluorescently labeled so that when we look at it under a confocal fluorescent microscope, we will actually be able to visualize how chromosome 15 is positioned within the cell of Bloom syndrome cells. And this is what I would like to introduce you to, which is my data here. And it is a Bloom syndrome fibroblast cell in the stage of metaphase. And I know that this cell is in mitosis because mitotic cells are having that DNA condense into those X chromosome structures, so they are tightly wound up and they fluoresce much more stronger in that DNA signal that is in blue. And in red, where we performed FISH, is chromosome 15. And what we do with chromosome 15 is that we will generate surfaces through an unbiased computerized program. So what they find is that if chromosome 15 is close to one another and it's suggested of pairing, what the computerized program will do is render it as one surface, and that's how I ultimately determined that chromosome 15 is paired. In contrast, what we could also find is that chromosome 15 is spatially separated, and what the program will do is render them as two separate surfaces. And so for my data, I found that chromosome 15 does abnormally pair within these Bloom syndrome fibroblast cells. So I would like to take a moment to just acknowledge my data. Understanding how chromosomes organize within the cell can give insight into how diseases within humans manifest themselves to the phenotypes that we see, specifically in Bloom syndrome. So this is why this project is significant. I would like to conclude my presentation by once again thanking you all for letting me be a COPLAC presenter, and acknowledging my principal investigator, Dr. Lisa Hua, and my graduate student mentor in the lab, Jem Morales, for their tremendous support and their mentorship, as well as Gabriel Quintero Pancarte for his technical assistance, and the rest of the lab for their kindness and support. Thank you so much.